Good morning, friends. Today we are still at topic 6 and we are going to look at how guard cells open and close. Alright, so before we move on, we have to recap the summary of our previous lesson. So before this, we were looking at how water moves up the plant. And there are three main forces that are needed to move water up the plant. The first one is called root pressure. So during root pressure, water moves from the root hair cells to the xylem vessels, which is at the center of the root via diffusion and osmosis. And this gives a pressure that forces water from the roots to the stem. The second type of force is called capillarity. And capillarity is basically the tendency for liquids to travel upwards through a very narrow tube. And the xylem vessels itself, it has a very narrow lumen, and therefore capillarity would allow the water to move up a distance at the stem. And the third force is called transpiration pull, which is a force that draws up water from the stem to replace water lost by evaporation. This causes a continuous stream of water to move up, called a transpiration stream, and this is driven by the process called transpiration. So we had to define transpiration, and transpiration is the evaporation of water from the surface of mesophyll cells, followed by the loss of water vapor from the leaves and through the stomata. So this is our future learning objective, but we are not going to cover any of it today because if you have a look at the learning objective, especially here, that the stomata is involved in this learning objective. And even this learning objective over here, effects of air, temperature, humidity, light intensity, and transpiration rate, these all are actually affected by the stomata. And even wilting has something to do with the stomata pores being open or closed. So our learning objective today, we are going to pretty much look at this learning objective, but we're not answering any of them. So we're just going to have our step to success that I have personally made for you guys. So if you are using your Cornell notes right now, you can use the step to success as your learning objective and the questions as your guide. So the first step to success for today is to state the function of guard cells. Secondly, to state how stomatopore opens. And the third, to state how stomatopore closes. So we are mainly focusing on guard cells, their function, and what is the process of opening and closing the stomatopores. So the guard cells. So what is the guard cells? So guard cells are a pair of specialized lower epidermis cells. So this is a photo from a SEM, scanning electron microscope, and you can see this is actually from the bottom of the leaf. Over here we have a pair of guard cells, and guard cells are really distinct. They're very special, and you can see that these are all guard cells, and the black color right there is the stomatopore. Just like you, if you do, I have pores on my skin, and when you look at pores, there is kind of like a hole there, and if you can imagine, the stomatopore is an opening, and it actually connects to the intercellular airspace of the inside of the leaf itself, so that gases can actually move in and out of this pore. But over here, if you look at those like jigsaw puzzle pieces, those are actually cells, and those are the cells that are surrounding the guard cells, and these are actually the cells of the lower epidermis. They do not have, especially this one, it doesn't have any chloroplasts. I think these are just chloroplasts that are found. I think they scanned it from other cells, but however, lower epidermis does not have any chloroplasts except for the guard cells. And you can see how different the lower epidermis is compared to the guard cells. So what are the functions of the guard cells? There are two main functions. The first one is to control the size of the stomata, which is the opening and closing of them. And secondly is to carry out photosynthesis. They do have chloroplasts for that reason. Okay, so how do guard cells open and close? So during the day, guard cells carry out photosynthesis, and this is because they are the only ones here that actually have the chloroplast. So during photosynthesis, sugars form at the cytoplasm of the guard cells, and this then lowers the water potential. If you guys remember, so if there is sugar concentrated in a solution, and if you remember, the cytoplasm is basically a solution, it is jelly-like fluid, and when you have more sugar in there, that means that it's more concentrated with sugar, and therefore there is, compared to water molecules, the water molecules would not freely move, therefore we say that it has lower water potential. So these would have lower potential compared to these other cells surrounding them, which is the lower epidermal cells. So these guys would have much more higher water potential. So having this potential difference, then you know what will happen next. Next is that water from surrounding epidermal cells will move into the guard cells by osmosis. So because of this this osmosis water potential gradient, then water will move into the guard cells via osmosis. So what happens next is really important because it's related to the structure of the guard cells itself. So we know that they are bean-like shape, but if you look at the drawing right here, this is legit on purpose. The fact that the walls, the cellulose cell walls of the inner part right here, this one side that is closer to the stomatophore, is actually thicker than the other sides. So guard cells have thicker cellulose wall, one side of the cell, the side of the stomatophore. When guard cells become swollen, aka turgid, this will become more curved and actually pull the pore stomata open. So if we have a look at this video, so you can actually watch this video at this link. I'll link in the description box below. And I'm not going to play the voice for this because it's really hard to record the voice because it only comes from my mic, so I'm just going to narrate here. So what this guy is doing is that he has a balloon. Okay, just so you know, these are the captions that are automatic by YouTube. So instead of turbo, he actually meant turgor pressure. Okay. So this balloon represents the guard cell, and if you can see that when he actually blew up the balloon, that shows the guard cell being swollen, but over here that it has a little bit of a curve, but not too much, right? So this is kind of like representing without having a thicker wall at one side, that it is a more like uniform shape. So what happens, what he's going to do is that he is going to add some tape right here. So when he added the tape, this represents actually that thicker cellulose cell wall on one side of the guard cell. And let's see when he starts to get the balloon to make it more swollen. And you can see that when he blows up the balloon, then you can see that it becomes more curved because that the wall over there is much more thicker. So because of this special difference in size, then that is how the guard cells can actually open and close the stomata pore. Okay, so what are we seeing here? So during photosynthesis, so in the morning, guard cells will produce the sugars. So this lowers the water potential of the guard cells, and water would enter the guard cells by osmosis, and this would cause the guard cells to expand and become turgid. And because of that difference in thickness of the wall of the guard cells, the guard cells will then curve up even more, therefore stomata pore will open. So as a result of osmosis, the guard cells become swollen and turgid. This is because of water actually entering from the surrounding cells and into the guard cells. So because of the thicker walls at the side of the stomata, guard cells curve up and stomata pores will then pretty much open. Okay. However, during nighttime, because there's no light energy during nighttime, unless you actually give the plants light, so guard cells wouldn't photosynthesize because no sunlight. Sugar will then be used up. So the sugar over here will actually be used up during respiration. And so when you don't have sugar anymore, then that will change. Well, sugar, okay, yes, it will be in 48 hours. If you keep it in the dark, then all of the sugars will, so all of the starch will become converted into sugars, yada, yada, yada. And so what happens is that this would also change the water potential of the guard cells. So at night, the water potential changes because there's not much sugar being made. I mean, not at all are being made because there's no sun. Okay, I'm sorry, I'm rambling. Anyway, so guard cells gain in water potential. Less sugars are dissolved in the cytoplasm. Therefore, water will then, so this one will have higher water potential now. So water will move from the guard cells, to the epidermal cells via osmosis this time. So because the guard cell loses water, that means that they will then lose turgidity, and stomata pore will then become closed. So that is why the stomata pore closes. So that is the end of the explanation, and let's go back to our step to success. So firstly, state the function of guard cells. Firstly, it's to control the size of the stomata, opening and closing of it, and secondly is to carry out photosynthesis. Second, state how the stomata pore open. So the steps here is that during the day when there is light energy, photosynthesis causes the guard cells to produce sugars, and then this will then lower the water potential of the guard cells. After that, water will then enter the guard cells via osmosis, causing them to expand and become turgid. Guard cells will curve up even more, therefore opening the stomata pore. And how do they close? This is at night when there's no light. So photosynthesis stops and sugars are used up for respiration. Therefore, this increases the water potential of the guard cells. So once the water potential increases compared to the surrounding epidermal cells, water will then exit the guard cells via osmosis, and guard cells will then lose its turgidity because they lose water, and the stomata pore will then close. So that is the end for today, and I'll see you guys next time. Have a good day and stay safe, everybody. Thank you.